Hello students, welcome to lecture 3 of week 12. So this is also our last lecture. So in the previous lectures we have looked at what are metagenomes and like the common ones including the human gut microbiome, environmental metagenomes, right? So in today's lecture we will look at the taxonomic composition of metagenomes. Okay, so basically it is, you can say, like microbiome. So here we are more interested in just the identification of species, right, and not the like the genome or the fragments of genome like metagenome that we call. Okay, so basically we want to classify or identify the genomes which are there in an environment. Okay, so instead of going for like detailed function analysis, we just want to know what are the communities first. And many times when we know about the communities, we can use the additional functional tool like Picarist to identify the functions also if we know the community well. Okay, so basically for classification we have to do the taxonomy, and to assign a taxonomic group or name to a species, we have to first see that where or what are the features of the species and where it belongs to, right, using the molecular markers that we have. Okay, so basically using taxonomy, ultimately we will be assigning a species a binomial nomenclature based name that will be composed of the genus name and the species name that is written in italics. Okay, so this is a common traditional system of modern system of basically assigning names, because it is more scientific, because you identify something. If you assign a genus and species name, then somebody else sitting at a very distant location can also give the same name and use it in a that bacterial species for their research. Okay, so it avoids ambiguity. So basically microbes are everywhere, including, it could be an open environment like lake, open ocean, okay, volcano, it could be like beneath the sea, it could be your oral cavity or your gut or skin, right, or a microbe can be present in the like other animals also. So basically if you want to identify them, it is a difficult task, and in metagenomics we aim to sequence each and everything which is there. So before that, if we have a good idea of what species are there and how many species we are dealing with, it helps. And basically the culture based techniques, which were the traditional methods like looking under the microscope or identification by gram staining or culturing, that is not possible here, because 98% microbes cannot be cultured by traditional methods, and we have to employ metagenomic based approach that we have also discussed in the previous classes. So basically, as we mentioned earlier, metagenomics helps you to understand first of all like what is there, okay, how much is there, and what are they doing in that location. So basically the identity, their numbers, and also their function. Okay, and for which we use the sequencing as the main approach. Okay, so basically with sequencing there can, so basically in metagenomics there can be two types of analysis. One is aimed as just the identification based on the 16S sequence, where we can use a particular variable region or the complete 16S, okay, or and amplify this, right, or make a clones of 16S ribosomal RNA if you want to construct a library of that, and identify the operational taxonomic units that we will see today. Okay, or basically we can break down the genome into multiple pieces or fragments. Each color is like one fragment from a single genome. Okay, so like you have different color bangles, you throw them, they will break in different ways. Okay, and this way, like you have to, the idea is to the color will tell the piece, color piece will tell that color existed or that genome existed, and their numbers will tell how many are there, right. And the idea is to reconstruct the pieces by, if you want to go for like detailed genome sequencing, right. So in metagenome we are mainly dealing with fragmentation and then of the genome, and not any marker sequence. Okay, so this part we have discussed in the previous lecture also. Okay, and then going for the function analysis. So in this case we can also identify what species is there and their function, whereas in this case we can only identify the species name. Okay, or many times it doesn't reach the species name also, but it tells you like overall diversity or what is present in that environment. And later on you can also connect with the function analysis if you have used a third party software like Picarist or something. Okay. So, and then basically either of the two goes to the computation analysis, which is a detailed comprehensive method. Okay, so it all starts in a metagenome by first of all taking the raw data which is you have generated or you are taking from some of the study. Okay, and then you have to pre-process or quality filter the data. You have to remove the barcode sequences, adapters, primers if they are rare. That we also did in the case of genome analysis. Okay, and then in metagenome, like if it is a human or host associated metagenome, you have to remove the host reads. So, for example, human or any other. Just for example, I have given human. Okay, then the remaining species which are like non-eukaryote or non-human, right? So we have to do the taxonomic assignment of that. Okay, so for that we can use several tools are there like Metaflan4, Kraken or Kaiju that we also discussed in the previous class. Okay, and then we will go for the preparation of abundance tables and the downstream analysis that includes their calculation of abundance, estimation of diversity by using various statistical methods. Okay, so for the taxonomic analysis, 16S ribosomal RNA is gene is the major molecular marker because first of all it is present across bacteria or like prokaryotes, and second it is present as well as it is variable in those bacteria. Okay, so you will always detect and it will be slightly variable, which is an indication of what species we are dealing with. Okay, so like this is a and this is a like 1.5 kb gene. Okay, 1500 base pair gene. Okay, and basically the structure is it has the constant regions, the green color one, and the variable region V1, V2 and up to V9. Okay, so 10 constant region and 9 variable regions. So variable or you can say hyper variable regions will show a lot of variation between different species, genus, phylum, okay, like that. And the constant regions will remain more or less consistent or constant across genomes. They may show some variation like at a very long distance, okay, like between phylums and like that, but more they will not confuse you in the identification of genus of species, right? So you can use either the complete 16S ribosomal RNA or you can use some part of it, okay, for the computation analysis. Okay, so basically this 16S ribosomal RNA is a part of RNA operon which is there in a cell. Okay, so basically in a circular genome if we talk about, okay, there is an operon, okay, ribosomal RNA operon consisting of 16S ribosomal RNA, 23S and 5S. Okay, and basically this operon contains your gene of interest, that is 16S ribosomal RNA gene. Okay, and there could also be multiple copies of this operon in a cell, in a genome. Okay, so it is not that basically you are dealing with just one copy, so you also have to normalize accordingly for the genome. For example, E. coli may have seven copies, so you will get 7 times more amplification or markers for this for the from the E. coli. So you have to normalize also for this species. Okay, so basically this 16S is very good sequence because it is present everywhere and has variable as well as constant regions. So if I want to amplify this region which is large enough V3 region to identify a species, so I can design primers from this constant region, okay, to amplify this part V3, right, and it will show me and so it will be a good enough region to be used as a molecular marker to identify the different species, right. And this constant regions can help me to because primers can go and bind in that constant region because it will not vary much, and it will be help me to identify the V3 region or amplify the V3 region, right. So this V3 region is generally used, or a combination of V3, V4, even longer region. So basically it all also depends upon the sequencer that you are using, right. So if you are using going for 150 base pair read, 150 base pair paired and read, I think V3 region is better. If you are looking for MySeq 300 base pair read, 250 base pair paired and read, so you can go for the V3, V4 region combination. Or if you are doing Sanger, then basically the complete 16S is fine, because from one side you can generate say easily 800 base pair fragment, okay, forward, and from reverse also you can generate the same, okay, approximately 800 base pair. Okay, so basically with a 100 base pair overlap you can identify the complete 1.5 kb 16S ribosomal RNA chain. Okay, but that will be like slow and you have to do one by one. You should have the clone in hand, sufficient quantity of DNA, whereas in metagenomics we go for the entire community approach, so we don't go for like typical single 16S ribosomal RNA that you do when you isolate some species from that metagenome and do further analysis. Okay. So the primers for V3 regions are like these two which are mentioned here, 341F or 534R, okay, forward and reverse. And similarly with the complete 16S you have to pick one primer from the start like 8th position or 27th position, and one from the end 1492R, because this way you can cover like almost complete 1.5 kb gene. Okay. So basically the same way you have to first of all clear the data, and then so basically after the amplification you have to clean the data. Okay, because the raw data may contain adapters, barcodes that we have discussed earlier also, and the low quality bases, because these low quality bases may interfere with your 16S ribosomal RNA or V3 region identification, because there also you expect some changes in between the species, and if those changes are due to errors, then basically it will cause further errors in your analysis. Okay. So QIIME is one of the pipeline that we use for this purpose. Okay. So basically data comes demultiplex. So demultiplexing means because you are dealing with maybe multiple samples. So in a lake also you are collecting, say for example you want to identify all the bacteria in a lake. So you may take sample from multiple locations in that lake. Okay, so from one lake there are 10 different samples, and your sequencer can produce a lot of reads. So you don't overkill or do like too much sequencing of just one sample. So you can mix all the samples together, but before mixing you have to put a barcode to each of the sample, so that all the reads will contain that barcode at the front. Okay, so that you will front or back, so that you can segregate the sequences on the basis of barcodes, and you after adding the barcode you mix all them together, sequence together, and then demultiplex those reads into 10 different sets, right? So that is demultiplexing. Then adapters and are removed, QC is done, like you have to identify the reads which are above a particular threshold in terms of Q30 or Q20 that you are using. Okay, then basically removing the noise by like human contamination or other host contamination, and go for the taxonomic analysis to identify the taxonomy, differential abundance, and other statistical analysis. Okay, so QIIME is one of the pipeline for this, and it is a standard pipeline that is evolved very nicely recently, and providing new and new additions to it. Okay, so it's a good pipeline for the complete 16S ribosomal RNA based analysis. Okay, which is also we call Amplicon based analysis, because you are amplifying something, a part of the DNA, that is a gene, selected gene or its region, and basically Amplicon sequence variants you are using ASVs for your analysis. So it can be found, the paper can be found here and published in Microbiome. So the QIIME can be downloaded from this site. Okay, and Conda is one of the installers for QIIME. So Conda installation we have already done earlier. So today I will tell you like how to do a quick installation of QIIME. Right, basically all software have similar ways of installation, and these days like Conda, Mini Conda, these have emerged as very nice installers for that provides a good environment for the installation and easy installation. Okay, so you will download in the Conda environment the software. Okay, and by using the command wget, and then basically you will create the environment and install. Right, okay, so this is the file you downloaded. Right, and then activate it. Okay, and then for any further instructions or parameters we can do QIIME help. Okay, so these are the various options available in QIIME to calculate diversity or features or basically composition. Okay, So you can use combination of these options or any of these options for further refining your command line for the QIIME analysis. Okay, so basically QIIME is aimed to identify the operational taxonomic units or OTUs, or even like these days it's called Amplicon sequence variants, ASVs. Okay, so what is an OTU? So basically what you started with, you collected sample. Okay, sample contains multiple types of bacteria. Okay, right, in multiple copies, right, because in a lake it is not that one bacteria is present once. Okay, there could be like thousands or even lakhs of bacteria and multiple copies. Okay, so basically by using the amplification 16S based primers, you are amplifying a particular region of 16S and you will be getting small sequences. Okay, right, from all of these genomes together, and you don't know what sequence belong to what genome. So basically you have to segregate or bin these sequences based on their composition, or to identify like the genus or species from those sequences. So QIIME will basically segregate those sequences on various ways, we will see later. Okay, there are 3 different ways: de novo, close reference and open reference, OTU picking. So basically idea is to segregate them into, for example, this piece and this piece are showing a above threshold identity. So these two, okay, will be clustered together, right. If this one and this one are showing too much identity, okay, and they basically, and that means that they are coming from the same species. Okay, so basically they will be segregated again separately, and this way you will segregate all the species. So these are operational taxonomic units, means that they should be coming from the same taxonomic group. It could be a genus or a species or a family. Okay, and because they are show a high level of sequence similarity. Okay, and generally 97% identity is the criteria that we use for clustering these sequences, right. So it can be done either de novo, you don't have a reference database and you want to do ab initio clustering. So you will check or align each sequence with each sequence, and those which are matching to each other will be segregated or separated, okay, to form different bins. Okay, and each bin will be one taxonomic unit. So you will identify a representative sequence from each bin, and that can be the representative sequence from that taxonomic group, because all other sequences in that group resemble this with very high identity, and preferably you will choose the longest sequence from a particular group. Okay, so if you do not have a reference database, you will prefer the de novo OTU picking or de novo clustering, right. If you have a reference database such as RDP database or Green Genes, right, so you will prefer to, or basically you can construct your own database also, then you will prefer to align these sequences against a database of 16S sequences, or basically the particular hypervariable region that you are dealing with, and see that what species it belongs to. So that will reduce your job, because you have sufficiently identified with sufficient identity the species which are present in that sample, right. So but still there will be some species which will be left unassigned, because they could be not known so far, very unique or novel and not present in the database. So this close reference, if only close reference is used, then basically it may lead to a lot of data depending upon the location or the metagenome you are talking about, environment. It may have so many new species, and that will not find a match in your close reference OTU picking, because you are using a reference database for the identification of OTUs, right. Therefore, a combination of the two is used. In that basically we can do first a close reference where we can align against a database, okay, identify the clusters which are known or basically coming from known species or genus, right, and the remaining species we do de novo clustering, so that we can identify the OTUs which are novel to that sample. Okay, so that is why this open reference OTU picking is better way in this way, so that you don't lose any data, and you also identify that what fraction of your genome and is containing novel sequences and basically what is their distribution and or basically the abundance, right. So basically RDP classifier is also there a tool. Silva is one database, GreenGenes is another database that can be used as a reference database for the close reference or open reference OTU picking, and these are constantly updated, and you can also add your sequence of interest in these databases if you think that those are not present, okay, so that it can also be assigned to the taxonomic groups, okay. So this is Silva, okay, it is present here, okay. This is the RDP classifier. So all these databases are published. You do Google search, you will find them. I am also showing it here that from where to download and use it, okay. Then this is GreenGenes 2, okay. So basically 1, 2 versions are there. So this is the second version which I am showing here, the latest one, okay. So all these are reference databases for the open or close reference OTU picking, okay. And then once you have classified the sequences into OTUs, the job is to construct a phylogenetic tree based on the distance between them, okay, and so that you can know that what species belong to what group, okay, or clade, and generally it will be taxonomically also closer to the genomes it is clustered together or in that particular clade, okay. So if it is a new genome and you are or a new OTU and you are able to classify that with a particular like say E. coli or say Prevotella, then you can know that this could be some of the species or new species from that group, okay. Then in the next step we are interested in doing some statistics to identify that what is the diversity in your sample, how many sequences or how many species are there, and whether they are evenly distributed or okay, what is the difference between the two samples in terms of the species. So alpha diversity is one important matrix which evaluates the intra-sample diversity, okay, that how much variation is there in a single sample, okay, like okay. So basically you can compare like samples from two different locations, okay, or you can compare like within the samples how much is the diversity. So basically for alpha diversity the the information that you need is species richness, that how many types of species are there in your sample, okay. So that will tell, because it could be that one sample is having like a lot of species, other sample is having very less species, okay. So basically different body sites, for example, before comparing the two body sites you can say like how much intra sample or multiple samples from that body sites, whether they are showing a lot of intra sample variation, right. Okay, so basically Shannon index takes care of both the species richness and evenness. So evenness means that it could be like there are multiple species present, okay, but whether they are evenly distributed or not, okay, whereas the species richness tells that how many types of species are present, okay. So both together can tell you that overall idea of the diversity of microbes in an environment, okay. So like we'll not go into details of this, we have already covered this in the metagenome analysis part, okay. So basically this species richness, evenness and together the diversity tells is very important matrix. And then you should do the beta diversity analysis, that is between the samples, it could be from two different locations or okay, or two different individuals, like what is the diversity between them, okay. It could be healthy disease comparison, right. So this will evaluate that how different the species composition and their basically in terms of richness and evenness is differentiating between the two, okay. And further we can also see whether the origin of bacteria, whether they are more taxonomically close or very different in the two locations, okay. So basically for example if I do like the beta diversity from multiple sites in body, you can see that you are doing clustering based on diversity. So you can see that basically the samples or the microbes from say oral cavity or gastrointestinal tract, urogenital tract or skin, basically they were very nicely segregated. That means microbe distribution in these sites or diversity in these sites is very different, okay. Those you find in skin you may not be in the gut, or some may be overlapping, but mostly like they are different, okay. Okay, so unique frag distance is also one of the important metrics here, which we'll also take further in this lecture using a example also. But basically, once you have done, you are comparing between two locations, you also have to see that what is the origin or evolutionary distance between those, and that we can do by unifrac distance. Okay, so then principal coordinate analysis, which try to reduce the dimensions in your data, and from a multi-dimensional data, multiple samples, multiple locations could be there, right? And it tries to find the principal components that can explain the maximum diversity or in your samples. Okay, so basically it tries to correlate. So first of all, you have to have the you have to do the Amplicon analysis using QIIME, and basically generate the OTU table for which various samples. So these are the different, say, OTU groups, right, and their respective composition in different samples, or different numbers in different samples. Okay, so there could be some OTUs which are more frequent, some are which are less abundant. Okay, so for different number of samples, like 40 samples and 270 ASVs, or 270 OTUs, or basically you can say 270 species, or that kind of taxonomic groups were found. Okay, then you calculate the distance between them. Okay, and then plot that distance on the principal PCA plot. Okay, so you can see that it using the Bray-Curtis matrix, which basically evaluates the compositional dissimilarity between the two sides, okay, based on the counts at each side. So you can say that S1, sample S1, S2, S3, S4, they are separate from each other, where like these two are like relatively closer as compared to these 3 and 4. So basically it is trying to explain the dissimilarity, or in other way the similarity between sample and dissimilarity between the samples. Okay, and we have already done principal components when we are doing the R series of lectures, right, and also in the metagenomic part. So basically to run these codes, you will use R statistical package. It could be R Studio or the R command line, where basically you have the data, you will be reading the data from the table. Okay, and then using the appropriate statistical analysis method. Okay, take the data. Okay, and then basically plot the data. Okay, which we have seen in the previous slide. Okay, so I will not go into details of this because we already did it in the earlier classes, but basically R is the statistical tool package where you can do the plotting as well as do the statistical analysis. Okay, for example, this is the distance between the samples. Okay, right, and then we are calculating the dissimilarities and going to plot these using the PCA plot. So this is a PCA plot for, say, healthy and diseased. So you can say that there are some points which are, first of all, there is a good overlap between the sample, like, okay, so basically this is the overlapping region which is containing like almost all the samples, and it is not able to explain a lot of variation. That means the principal components is not able to explain most of the variation in the data. Okay, only 16% on the first axis, right, and only few points are segregated, but still they are very close in terms of the overlap. Okay, whereas in this case you can see that there is a clear segregation between the colorectal cancer samples, whereas the healthy samples, these were the samples of bacterial species from extracted from the fecal matter. Okay, so basically when CRC patients and healthy patients, you can see that there is a clear separation between the two, and these kind of and so this can help you further to first of all tell that basically the composition is very different between the two, or there are dysbiosis between the two, and also like some of these bacteria can also be used as a marker between to segregate the CRC or healthy in the future. Okay, there is a tool called Megan. Megan 6 is now there, and basically it is a automated pipeline, or you can also install or use it at their site to basically analyze your metagenomic data. Okay, so it will do the clustering, and also it will basically look at the functional analysis using interpro, seed, NOC, CAG, etc. Okay, and then make the PCA graphs and basically this kind of phylogenetic tree, where it will also give you proportion of different species, you can also compare multiple samples here. So Megan or metagenome analyzer is a very good tool that has been used in several studies for the metagenomic analysis. Okay, so basically in the 16S Amplicon analysis, this was a pipeline that we discussed. Okay, so once you have the clean data, you will identify the representative sequence from the OTUs that you have made and do the taxonomic classification of those, and then do the statistical and function analysis. So I'll give you example from one case study which will help you to connect with the earlier concept that we deal today. So that is the example of, say, hot springs from the central India. So this will also tell you how to start a project. So for example, there are so many hot springs in India, and like particularly central India, it has some important hot springs like Choti Anoni, Badi Anoni in Madhya Pradesh and Tatapani in Chhattisgarh. So where Choti Anoni and Badi Anoni had temperatures of like medium or medium level hot, like 55 to 61 degrees, right? Whereas Tatapani had very high temperatures, are like up to 97, 98 degrees centigrade. Okay, so you can extract DNA from those and do the either the function or taxonomic study. Okay, so basically what you will be doing is you will be collecting samples from there in a clean collection device bottle, right? And then you have to transfer those samples to the lab as early as possible. Until then you have to store the samples in preferably in -20, but if not possible then in ice packs, and you have to bring them very quickly to the lab. Otherwise what will happen is the diversity may change while you are transporting that to the lab, because you have changed the environment of that. The water will cool down and it may allow other communities to basically take this space there. So basically these sites are located in this region, and samples were collected from those. Some were having like 50s 40s degree temperature. Microbes are growing in that, and up to like 98 degree in Tatapani 1. Okay, where it is very difficult to imagine that microbes may be there, but still your microbes were found. Okay, so the first thing that you will do is do the physiochemical property analysis. So like temperature, what is the temperature and those samples, the pH, total solids, right, and organic matter detection. So depending upon the site and your hypothesis, or the area, or your objectives, you have to identify the physiochemical properties, right, so that later on you can correlate the abundance of species with that properties. So basically for the two different sites this was done, right, and you can see multiple samples were collected from the site, so that you will know that basically what is the inter-sample and intra-sample variation between the samples. Okay. So first of all you have to extract the DNA, but before that you have to remove the debris. Okay, so you basically filter, which is 1.2 micrometer filter was used to remove the debris, because in any water sample or this there could be so many other things like algae or some contamination. So those, at least the visible one, can be removed by the debris can be removed, because microbe size is much smaller than 0.6 micrometer or so. Okay. So basically you can easily remove by using this so all the debris will be on the filter paper, and the remaining particle will be collected, right, and then this filtrate is passed through a 0.2 micrometer pore size, so that the microbes can get trapped on that. Okay, and the remaining like smaller things, such as like it could be the viruses or those that will go through. Okay, and only the microbes will stay on the filter paper, and then you can keep that filter paper, preserve that for further analysis. You can do multiple extractions from these filter papers, and also you can divide and keep some for like analysis today, and later store some, so that you can again go back and redo the DNA extraction from those in the future. Okay, and then you can also identify like some of the genomes from there may be culturable, so you can also go for the culture based approach okay, to do the genome analysis of the genomes which may be present in that location. So for that you have to mimic the conditions of that environment, such as the pH, temperature that you have already recorded, presence of other solids, okay, or even like you can like use the same water that you have got, okay, remove everything from there, like all the bacteria, viruses, anything that or human contamination from that water, okay, again autoclave it and then use as one of the ingredient of your media, so that the species may get the similar type of nutrients that they were getting in the original environment. Or you can know that what were present, if that knowledge is there, you can use that it while preparing the media and including the other physical condition that you are mimicking, okay. So basically from these sites various 3 genomes were identified, isolated and identified, and went for whole genome sequencing, okay. Now again, like we have done alpha diversity and beta diversity and Shannon and rarefaction in the previous one. So again we will see these here. So the alpha diversity, you are trying to see the intra-sample diversity of species, and beta diversity, you are comparing the 2 samples for their relative composition and diversity composition, okay. So for these you will also be doing the rarefaction, okay. So that because rarefaction is important because it tells that many things, because sometimes the amount of data generated from 2 different sites may be very different. You are dealing with a lot of sequencing at one site and very less sequencing other site. So basically you can create subsets from the 2, right, including the equal amount of data, or using iterative subsets from the 2, so that if all the subsets are showing the similar type of distribution, then you will know that basically you have kind of normalized the data from the 2 sides. And also rarefaction can tell you that whether you have done sufficient sequencing or not, because if you are using say fresh water from say your tap, okay, the amount of number of species there, whereas in a lake or basically in a drain it will be very different, okay. So the amount of sequencing that you require for ocean or a lake or that drain, okay, sewage drain, may be much more as compared to the amount of sequence that you will require in the tap water, because tap water is basically should be free from most of the microbes, or only few should be there, right. Okay, so basically then basically you have to put a cutoff also, that how many species you will be plotting, because there could be like thousands of species, and it is not possible to view everything on the graph, or okay. So basically like greater than 1% abundance you may choose to plot this, for example in this study, right. So basically that way you are covering all the important species, or you can say like for sure the more abundant ones, okay, and see their distribution, right. Then apply the various statistical methods to look at the abundance between the 2 locations, and then construct the phylogenetic tree that we will see later, and also the unifrac distance tree to see that how the samples are related to each other in terms of the evolutionary or phylogenetic distance, right. And then also to identify the discriminatory taxa, that the taxonomic groups or microbes which are significantly different between the 2 locations. You can see like visually that okay, A is more and B is more in the 2 samples, like A species is more here and B species is less, or B is more here, okay. But basically whether that is statistically discriminatory also, that you can use by using some of the software packages such as Boruta and LabSe. Both are based on random forest and available in R, okay. So for example in the rarefaction you can see that if you increase the number of sequences say from 100 to okay this number, okay, you can see that initially with the increase in number of sequences or amount of sequence data or number of sequences considered to call the taxonomy or do the taxonomic analysis, you can see that there was an increase first, okay, up to this point you can say, but after that it became relatively constant. That means even if you do more sequencing, more and more sequencing, there is like very little or no chance that you will add a new species here. The their number diversity will remain almost same. So, you will not be able to identify more. Okay. Similarly, for the different sites, if you see like right, for example, here adding more sequences, basically this plot becomes flatter very soon, whereas here there was still some chance to add more species also, because it may have gone further and got stable later. So, but still basically you can see that it was not too much increase, and the amount of sequencing was sufficient. Okay. And basically based on various statistical and based on temperature, pH, etc., you can also see that whether the species from a location are clustering on the basis of temperature. That means at very high temperature and medium temperature, whether the two locations are also showing the same type of species or very different species, right. So, basically you can say high and extremely high were linked together, and extremely high and moderately high were linked together. So, basically you can identify the species that are significantly different, like this is significantly different, and those are plotted here. Okay, then the phylogenetic and functional similarity can also be measured in the different locations. For example, in terms of the text, so for example, this plot is for the taxonomic abundance of say different location like Tatapani 1, 2, 3, 4. Okay, so one was very different from the other three, and one was having the most highest or the highest temperature. Okay, and the other three sites, Choti Anoni, Badi Anoni, and like these. Okay, the cluster together. So, basically on the basis of unweighted unifrac, that means like all species were similar and no weight was assigned. Okay, so you can say that the samples were clustering on the basis of the communities, and that was also an indication of basically it also emerged that basically because of the temperature difference, these four sites were labeled or numbered, and the species also varied according to the temperature, and that was also reflected kind of in terms of their function analysis. Okay. So, in unifrac, basically if we go further, we want to create a using the phylogenetic distance between the species. Okay, we want to connect these species. So, for example, there are two communities which are identified by red squares and green squares. Okay, green circles and red squares. Okay, so this is their you can say their unifrac distance. So, here if you see these branches are colored differently, right, and they are not very different in terms of their evolutionary distance. Okay, so unifrac measures the amount of evolutionary distance between two communities by dividing the length of purple branches by the total branch length of the tree. Okay, so this way you can see that there was very little evolutionary distance okay, here in this case, whereas here you can say it is or purple labeled. So, okay, unifrac distance total by this purple color. Okay, so on the right it is showing that basically the two communities are maximally different as the lowest common ancestor is the root of the tree, right. Basically here their LCA you can see their lowest common ancestor is basically if you have two species. Okay, so and they are different from each other, you will go for the lowest common ancestor, that is at the genus level classification. So, if at the genus level the two species are two genuses, the genera that you have identified are very close to each other, you will go even at the lowest common ancestor, that is at the family level. So, in this case basically the lowest common ancestor was just here. Okay, that means these two species which are in green and red basically can be connected by this, which is belonging to the same genus perhaps, or the same basically above taxonomic group, whereas in this case they were all different and and could be connected at this level, which is at the root or basically at the phylum or at a very high level. So, these were very different from each other in terms of the phylogenetic distance, whereas these were very close to each other. Okay, and that is how like you can also see like in the different locations you will identify the microbes which are present and their relative abundance by using various colors, and you will find different type of microbes abundant in different locations, which I will not go into detail of this, but basically this picture tells that you have plotted the distribution of different microbes at different location and their relative composition, right, and which is giving you an idea about what species were abundant there and what could be their role in that location, that you can interpret using your knowledge and further reading. Okay, similarly you can connect, because you are taking samples from locations which have different physiochemical properties. Okay, you can also see the relative distribution in terms of the physical chemical properties, and using this heat map also you can see here. So basically that will display that microbe how much is the similarity, like, okay, and difference between the microbes in terms of their based on the physiochemical and the geochemical properties in that location. Okay, you can also try like a comprehensive taxonomic tree by combining all the communities having some relative abundance like 1% and above here, right, from the different locations, right. Then see like whether the abundance of any microbe can be correlated with the temperature. Like in this case you can see that okay, there were some which were showing the archaeal species showed correlation with the Tatapani because it was high temperature and archaea prefer high temperature, whereas the hydrocarbon degrading species were more abundant or less abundant in very high temperature region, but more abundant in the lower temperature regions. Okay, for example in this case the idea was to find the species that can do like hydrocarbon degradation, because basically in these locations hydrocarbon abundance was one of the feature, because the you can say the digging or the wells were bored to find the species that can basically degrade, or basically by thinking that it could be like potential oil reserve sites. Okay, so you can see that in the Anoni site there were species abundant which were involved in the hydrocarbon degradation, whereas in the Tatapani site, which was a very high temperature site, basically most of the functions the genomes were doing was mainly for their own survival, that is DNA repair and amino acid synthesis, DNA synthesis and like that. Okay, so these are some of the later interpretations or subjective analysis that you would like to do based on your knowledge or hypothesis that you started the project with. And later on you can further analyze like various, for example, hydrocarbon pathway emerged as, or hydrocarbon degradation pathways and the capability of microbes to do that emerged as one of the important outcome. So you can connect like what microbes are there in methane metabolism in the different sites, okay, particular microbe and its abundance in various sites, and similarly toluene degradation, these microbes were present and it was more in this side whereas less in this side. The circle and its size of the circle can tell about that. Okay, so this way you can do further analysis and bring out the microbes that can do the potential function that you are interested in and their relative abundance in the different locations. Okay, and then you can go even further and try to connect to the pathways level, that how the microbes would be involved in like the degradation of that hydrocarbon, like starting from toluene to basically ultimately how they are using those hydrocarbons for their survival, and converting the toluene to pyruvate, basically, which is basically the input for the TCA cycle, and further doing the entering the electron transport chain after the TCA cycle to get the energy. Okay, so basically in this lecture we looked at the taxonomic analysis, what are operational taxonomic units, and how we start like from the collection of sample to the taxonomic analysis and further the statistical analysis. And we also looked at one case study where we see the real application of the statistical test and made the interpretations based on the research problem and the sample that were collected from a particular location. Okay, so this is good for today. Thank you.